Okay, this is the study that we're doing based on what Wilma and Lee did as the first response to the oil spill in terms of community science. And the design was so good, we pretty much have adopted it. What we have here is a thing we call the Gulf Coast Health Alliance. We're looking at the risks related to the Macondo oil spill. And why are we doing it? We're looking at the long-term health effects of exposure to PAHs of the kind that are part of the oil, not from burning, especially from consuming seafood. The project has 4 pieces to it. The one we're talking about tonight is really number 2, although the actual collection is part of number 1. We're looking at number 1, and it's run by 2 doctors called Croissant and Emmett, Dr. Emmett, Dr. Croissant. It's a study of people that live on the coast that eat seafood, looking at whether or not they have PAHs and whether or not the PAHs are accumulating. It's also a study of communities in terms of the vulnerability as a result of the spill and other factors like risk factors that already existed from all the different things that happened here, because a lot has happened here since 2005. I'm new to the area. I've only lived in the Gulf for 10 years, but I live on Galveston Island, so I know what hurricanes are like now, but I didn't before. I've been over here because of Katrina and Rita, but I didn't live here. Now I realize that it really does have a big effect on people. And then the oil spill, that's another level of risk. And we're looking at also how communities can be resilient and bounce back from things like that, and ways in which they can be helped. Project 2 is the one we're talking about tonight, really. It's the exposure assessment. We're looking at the exposure to PAHs by looking at tissue of different seafood species, and looking at, and eventually looking at blood and urine samples of people. And we're going to try and use that, and this will be Dr. Gil Rowe from Texas A&M. He's a marine biologist. He's going to try and use the thing he calls modeling to model where, through currents, through patterns of different weather, through migrations of species, how the oil, the PAHs, can go throughout the system, the food web, the ecosystem in the Gulf. That's what the inverse modeling is about. Project 3 is something that's never been done before. Dr. Penning, and he's from the University of Pennsylvania, is going to try and figure out, and I say try and figure out because sometimes these experiments are experiments, but he's going to try and determine the level of concern, in other words, the level past which something becomes toxic for the petrogenic PAHs. Right now, they don't have a level. It's never been done. So he's concerned about that. He wants to look at that. That's something we all need to know, because based on that, you figure out how much seafood you really want to eat. Okay? You can determine, you can eat this much, but don't eat, like they say right now, don't eat too much tuna because of mercury. Don't eat and where we live, don't eat too much speckled trout or blue crab or oysters because of PCB and dioxin. That's in Galveston Bay. That's not here. Okay, but that's how they figure that out. They have a level of toxicity and you don't want to exceed that. And then Project 4 is looking at things in the body that can indicate that you are at risk or you're under stress. So he's going to be looking at how exposure to these various things in the other projects affects your stress level, and they'll be using what they call biomarkers. Biomarkers are indicators like hormones, indicators like certain chemicals, catalyst reactions, things in your body that indicate that your body is under stress and is trying to detox. Okay, and these are biomarkers of that. And we'll be looking at that. And then we're going to talk now about what's been done so far on the seafood sampling. And this is with Wilma, Brian Padas, who's out there walking around, and myself. Okay, these are our 3 seafood sampling hubs, plus the Louisiana Environmental Action Network, which is our overarching sort of supervision of the sampling process. And Wilma went to each of the hubs. She goes to the hubs and shows people how to do this. The United Houma Nation is one. The Mississippi Coalition of Vietnamese American Fisherfolk and Families is another. The South Bay Communities Alliance, and that's in Mississippi. The South Bay Communities Alliance in Alabama is another. And you can see that the Mississippi Coalition covers the entire coast. The Alabama area is Mobile Bay, mostly, that area. So those are our project partners in this project. And our sites are in Louisiana, basically from Atchafalaya all the way over to Lower Plaquemines. That's pretty much everything. Mississippi is the entire coast, but we do have spots we're looking at. I'll show you the map in a minute. In Alabama, Bayou Loutre, Mobile Bay, and Dauphin Island. Those are the major places we're looking at. The species we're looking at are white and brown shrimp, blue crab. Right now, among finfish, it was speckled trout, but I think we're going to try and expand that because, for one thing, specks are hard. Well, they're either there or they're not when you go to catch them. And we'd like to get more finfish into this data pool. So we're thinking of expanding to drum, redfish, sheepshead, and maybe other species. We're going to ask you for suggestions tonight about this based on what people eat. And then oyster. The schedule is 2 times yearly. The number of sampling sites are 15 in the entire 3-state area. These are some things about the sampling, and I'm not going to read this whole thing, but I just want to focus on certain things. We're looking at PAHs again. We're looking at PAHs over time to see if they get the amount concentrated in tissue gets larger, stays the same, or drops. If it gets larger, we call that bioaccumulation. It's just accumulating. If it stays the same or drops, that's a different issue. We designed the site, the matrix we call it, of sites where we're going to be looking based on what we call proportional exposure. And Louisiana has more sites because Louisiana had more exposure. Mississippi has more than Alabama. Alabama has the least. And the last thing on this slide, I just want to mention again that the sampling plan, the protocols we call it, was developed from what Wilma is doing with lead in the lower Mississippi River deep. This is a rundown on the 7 sample points in Louisiana. And once again, when it comes time, we're asking you for some suggestions. If you could tell us places you think ought to be looked at, that's why we're here. We want to know that, because we don't know. But these are things we put together with our partners, and they're general, they're big, but within these big areas, there are places we need to go. And you can see in Louisiana, we start with Atchafalaya, Lower Terrebonne and Fouche, Grand Isle, Barataria, Lafitte, Dennis, Boothville, Point La Hatche, and St. Bernard. And then in Mississippi, we're looking at Bay St. Louis, Waveland, Pass Christian, Gulfport, Biloxi, Pascagoula, and Moss Point. In Alabama, we're looking at Mobile Bay, east and west in the bay, Dauphin Island, and Bayou Loutre. And here's a map of the entire thing. And the dots, if you look at the legend on the right-hand side, the dots indicate what we found. The colors they indicate data points. The colors indicate what we were sampling. Blue crab, brown shrimp, oyster, white shrimp, or speckled trout. The big dots with the crosshatch, that indicates that we have a seafood and human sampling hub, and we have one in Houma, and then one in Gulfport, Biloxi. Those 2 are combined. And then the simpler crosshatch is seafood only, and that's over in Alabama. We're not going to be looking at the human population there, just the seafood. Then this just fills in some of the names of the various places where we're going to be looking. And the next slide is just a closer look at the United Houma Nation sampling matrix. This is just Louisiana. We'll also be looking at some of the offshore waters, and the sample that I'm going to show you tonight came out of Area 13 off Grand Isle. Didn't come from around here because we got here too late in the year to get any shrimp. Close in, they were gone. And we got blue crab and we got oyster, but we didn't get any shrimp. So we got this shrimp, and it came off of a boat that had been out there for a while. And we got it from Mr. Blanchard over on Grand Isle. And I'm going down here to next steps. High priority for us, we want to get samples in Barataria and Lafitte, St. Bernard, Venice, Bluefield, Bay, St. Louis, and Gulfport, because we didn't get anything this time around from those areas. So we're going to look at those, hit them hard, see what we can get, and we're going to cover everything again. But those areas are blank at the moment, just because we weren't able to do it. And we're working on getting the analysis of the marine samples started. I'll show you how that's being done. Now, this is a complicated thing. I don't fully understand this myself. I don't do this. This is happening in the lab. It's part of Project 2. But this is a recipe in how you prepare a sample. The chemicals you add to the sample, and then you are, of course, grinding it because we gave whole crabs, so whole crabs being ground up. Things you add to it, you shaking it, putting it in a centrifuge, which swirls it around at very high speeds, and then putting it in the machine, this machine, which is a GC-HARMS, and it's basically going to take a picture of the composition of what's in what we're looking for. Okay, you're going to get a snapshot of the chemicals that we're looking for in the sample by heating it, bombarding it with ionized radiation, and looking at the things that are available in a snapshot, and it's based on the weight of these things. It's called a mass spectrometer, looking at the mass. How much does it weigh? Some of these are light compounds, some of them are heavy compounds. These are the PAHs we're looking at, and you can see a lot of these names were in Wilma's presentation, and they're the same thing. There's a few more of them here, though. We're targeting 42 of them. These are petrogenic PAHs. I'm not going to go through the list. And now let's look at a sample, two samples actually. We're using Galveston shrimp, oysters, blue crab as a control.